which has a 0.7 to 4% carrier prevalence in the European population and leads to a 2 to 3x risk of clotting. Unsurprisingly, neither of these mutations were present. I say unsurprisingly because both variants are virtually non-existent in African descent populations. So, we decided to pinpoint what mutation might be responsible. The first thing I did is look at the number of heterozygous variants shared by the family because the disease process suggested a dominant mode of inheritance. There were over 13,000 variants that fit into this category. Next, we decided to focus on genes with biological relevance, genes that had been shown in the literature to have been implicated either through rare variants or families or larger studies to be involved in deep vein thrombosis and pulmonary embolism. We had 9 variants that fit this bill. From there, we decided to focus in on variants that changed the amino acid because these variants are usually the most damaging. And finally, because this is a disease-causing variant, we were looking for something that wasn't at high frequency in the population. Based on this, we had one candidate variant to look at, which was a variant in protein S. Protein S is incredibly important for the clump chomping up process. When you have the clotting cascade come in, you have a host of factors that are working together to build that clot. And what protein S does is it comes in with other factors and forms this complex that breaks up your clot. And so if it's not functioning properly, you cannot break up that clot properly. Our mutation was a valine to methionine change in a functionally important region of protein S. Using algorithms which predict whether or not a nucleotide change is harmful, polyphen and CYP, both algorithms showed that this mutation was predicted to be damaging. And the literature shows that other protein S mutations, which are usually Europeans, are generally familial and lead to a 20x greater risk of deep vein thrombosis and pulmonary embolism. At this point, based on the genetic data, we went back to the individuals, the clinicians who were in charge of the family, and asked if we could get any available laboratory data that might corroborate our findings. We found that the 3 family members that we had sequenced all had clinical evidence for a free protein S deficiency. And this clinical evidence helped sort of make us feel more confident about what we had found. The next thing that we were interested in is figuring out whether or not this was a private variant. As I had mentioned before, in European populations, protein S mutations tend to be rare and familial. And we wanted to know if that was the same case here. So, we looked at a database of mutation frequencies called the Exome Variant Server. In 4,400 individuals of European descent, we found the mutation to be virtually absent. In over 2,200 individuals of African American descent, we found that the mutation happened in 1% of individuals. We then decided to look in the phase 3 of the 1,000 genomes to see if we see this mutation in other African descent populations. And indeed, we did see this mutation appearing at a frequency between 0.4 and 1.4 in populations that have descended from West Africa. So, now that we knew that this mutation actually does appear in the population, the next question was, is it actually a risk factor in that population? So, what I did is that I collected samples from Florida, Chicago, Vanderbilt. We got every available sample of African Americans who have a clinical history of clotting, and we genotyped them at this protein S locus. We had 315 individuals who were African American descent with DVT-PE, and 380 individuals who are our controls who had no history of DVT or PE. When we looked at the data, we found that the protein S mutation was enriched in the cases. The frequency of the mutation in the cases was 2.2%, and in the controls was 0.65%. Using a Fisher's exact test to test whether or not these allele distributions were statistically different from one another, we had a significant p-value and an odds ratio of 3.4. Given the incidence of this disease, the odds ratio is pretty close to the relative risk. So you could say that if you have this mutation, you are about 3 times more likely to fall into the disease group than in the control healthy group. Based on this information, I concluded that this mutation, this protein S mutation that we had found in the family and had clinical evidence for in the family, was also a population-specific variant affecting clotting risk in African Americans. And as far as I know, it is one of the first population-specific risk factors for clotting in the African American population. Now, I would like to move on from clotting to its treatment. And I would like to talk about genetic risk factors affecting achieving therapeutic dose of warfarin. So, if you have problems with clotting, what ends up happening is you get put on long-term blood thinning treatment. Warfarin is commonly prescribed in the United States. There's over 30 million prescriptions a year. It is one of the cheapest pharmaceutical options. There have been next-generation drugs that have come out since, but because of their price point, warfarin still remains one of the go-to drugs. The problem with warfarin is that it has a narrow therapeutic index and a high inter-individual variability in dose. What does that mean? That means that 2 people might need wildly different doses of warfarin, but you couldn't really tell with just looking at them. So, Andre the Giant might need 10 milligrams a week, even though he's huge. The Sicilian, he looks quite shocked when he sees his dose, might need 100 milligrams a week. And Inigo Montoya is an average 35 milligrams a week. Quite inconceivably, the way we currently dose warfarin is through guess and test. So this is sort of a rough representation of what might happen. I know the protocols are more complicated than this, but day 1, you administer a dose of warfarin. You come back a few days later and you measure the blood clotting. At that point, you still see that the person has too much clotting in their blood, so you increase the dose. Then, you come back a week later, and we measure your clotting again. Still, too much clotting, so we increase the dose. Then you would come back the next week and measure it again, and at that point, uh-oh, you have not enough clotting, the dose is too high, we should decrease the dose. As you can imagine, with this sort of regimen, what ends up happening is that you have a lot of opportunity for adverse events. Because if you dose too high, you get bleeding. If you dose too low, you get clotting, which is exactly what you were trying to avoid in the first place. And not surprisingly, because of this, it is responsible for 1/3 of the adverse drug events in older Americans. So, what can we do to try to be better at predicting what the warfarin dose should be? One thing that has gained some traction is using genetics. The field of pharmacogenetics is basically using genetics to make informed decision about your drug choice and the drug dosing. And we have known through research that genetic variation in the drug target gene, VKRC1, and variation in the metabolism genes, including CYP2C9, can affect your warfarin dose. So you can imagine that if you have a mutation in the target, maybe you need a higher dose of the drug for it to work. If you have a mutation in the metabolism and you can't get rid of it as well, you might need a lower dose for the drug to work. In work that I'm not going to go into great detail to right now, but you are free to talk to me about it later, I showed that if you aggregated the genetic variation along the metabolic pathway, all the different genes involved, and created this aggregate score, you could use this score as an additional predictor for predicting warfarin dose. In 2009, there was this wonderful paper that came out trying to estimate warfarin dose using clinical factors such as age, weight, other drugs that you might be using, and genetic factors. Of course, This study included mostly Europeans. After that, there were some clinical trials that tried to figure out how useful warfarin pharmacogenetics was in predicting warfarin dose. One study, which compared a pharmacogenetic algorithm to a clinical algorithm, which is not the current standard of care, found no difference between the two. But the second study, which looked at the pharmacogenetic algorithm versus the actual current standard of care in the UK and Sweden, found that the pharmacogenetic algorithm was significantly better. Unsurprisingly, given my talk, most of the subjects in those studies were of European descent. There were 275 individuals of African American descent in one of those studies, and it was particularly highlighted in that study that pharmacogenetics did worse in African Americans. Now, why might that be? In a letter to the New England Journal of Medicine, I described the reason that this study saw worse performance with pharmacogenetics in their African American population. That study included 3 variants for their algorithmic prediction. CYP2C9, which is a metabolism variant, star 2, CYP2C9 star 3, another metabolism variant, and VKRC1, which is the target of warfarin. As you can see, these mutations are highly enriched in European populations and appear in African American populations at a much lower frequency. And this statistical test that I used basically demonstrated that there was a statistical difference in the distribution of the mutation between Europeans and African Americans. So that the reason that this algorithm that was using these variants was performing worse was because you don't see these variants as much in African Americans, so they're not as predictive. I was interested in figuring out what was the undiscovered variation affecting warfarin dose in African Americans. So what I did is I took a distribution of African American individuals on warfarin, and I selected individuals who fell on the high and low side of the dosing spectrum. We exome sequenced 103 of these individuals. And then we created a model to see if we could find any new variation. We used the clinical and genetic variables that were already known to be helpful in predicting warfarin dose because we wanted to just find new variation. And we made sure to include variants that were specific to African Americans that we already knew about, such as CYP2C9 star 5, star 6,